Hopefully you can hear me right now. And I'm going to be doing a walkthrough of the DNA Microarray Lab for those who didn't have Flash Player. So this is going to walk us through the process of microarrays and how they show gene expression. This virtual lab is broken into 3 chapters. Chapters 1 and 2 will give you some background and review a couple important concepts that will help you understand the DNA microarray technology. Chapter 3 is the experiment. So we're going to start with Chapter 1, Genomics. Way back in the 20th century, a few years ago, scientists came up with many ways to study genes, mapping them, making mutations in them, cloning them, sequencing them, and analyzing the proteins they encode. Typically, scientists applied those kinds of studies to one or maybe a few genes at a time, but things have a lot of genes. Humans, for example, have around 20,000 genes. It would take a very long time to investigate each human gene one at a time. Wouldn't it be great if scientists could study many genes at the same time? Or even better, what if they could learn about every gene in an organism in one single experiment? That's a bold ambition, but that kind of thinking has transformed 20th century genetics into 21st century genomics. Genomics is a new word for a new science. Genomics is a way to study many genes, thousands of genes, or even every gene in an organism all at once. One proven tool for this mask is the DNA microarray. Chapter 2, Measuring Gene Expression. To see how powerful genomics can be, let's take a closer look at what makes various types of cells in our bodies different from one another. With a few exceptions, every cell in our body contains copies of each of our 20,000 or so genes. Different genes are turned on or off in different cell types. So how do we know whether a gene is turned on? Well, one way is to determine if it is working, turning out molecules of messenger RNA, or, as molecular biologists put it, if the gene is being expressed. For example, you might expect a muscle cell to make mRNA for various smooth muscle proteins, such as actin and myosin. But muscle cells should make no mRNA for the pigment melanin or the hormone insulin. If we could measure the amount of mRNA for every gene that is present in a cell or tissue sample, then we could make a genetic catalog of what sorts of biological processes are going on in those cells. And by comparing such gene expression profiles from 2 different cell types, we could learn what makes those cells different from one another. That's where DNA microarrays come in. Using DNA microarrays, researchers can pinpoint all the differences in gene expression between 2 different cell types in a single experiment. So let's run a microarray experiment. Before we begin our experiment, we will need a DNA microarray. Here's how to make one. Sequence the entire genome of the organism you're studying. Fortunately, this has already been done for the organism we're interested in, humans. Use massive amounts of computing power to figure out where all the genes are in your sequence. Do more computer crunching to design primer pairs so that you can use PCR to make copies of every gene. Using robots or a team of overworked technicians, make all of the primers and do a PCR reaction to copy every gene in the genome. Run quality control tests on all of the gene copies you make to find out which PCR reactions didn't work. Repeat steps 3 through 5 as needed to make copies of all the genes that didn't work at the last time around. Separate the double-stranded DNA from each gene copy into single strands. Use robots to place microscopic droplets of each single-stranded DNA sample into ordered rows and columns on a glass microscope slide. This is the microarray. Use more computers to create and manage a database to keep track of all the gene spots on the microarray. Do more quality control to make sure all the spots contain equal amounts of DNA. Don't worry, you're not going to be tested on this stuff. It's just so that you can understand all the work that goes into it. We won't be making our own DNA microarray today. We just happen to have one already made. It's right here in this lab drawer. Actually, preparing a genome-sized microarray is too big of an undertaking for most individual scientists in research lab. They usually purchase their microarrays from biotechnology companies, which are equipped to manufacture them in large quantity. There's a thing about gene chips. We don't need to worry about that. Let's take a close look at our DNA microarray. A microarray is made up of thousands of spots. Each spot contains multiple copies of unique DNA sequence, which corresponds to a single gene. As we'll see a little later on, this can be a handy tool for determining the difference between two cell types. Chapter 3, the experiment. We're going to use the power of genomics to answer a very important question. What's the difference between a healthy cell and a cancer cell? How might you try to answer this question? You could look at healthy cells and cancer cells under a microscope and compare their appearance. That's one way doctors try to diagnose cancer. But cancer cells don't always look different from healthy cells. And comparing how cancer cells and normal cells look doesn't really get at why they're different. So what does make a cancer cell different from a normal cell? Cancer is basically a disease of genes gone bad, in this case, gene 3. Many genes control the way cells grow, divide, and die. When those genes stop working properly, cell growth could spin out of control, leading to tumor formation and cancer. But different types of cancer are caused by different sets of genes. So to be able to better diagnose, understand, and treat cancer, it is key to know which genes in the cancer cells are working abnormally. In this experiment, we will use a DNA microarray to compare the differences in gene expression levels between cancer cells and healthy cells. The steps we'll follow are at the bottom of the screen. We'll highlight them as we go to track our progress. So far, we have a microarray. We're going to need some more equipment. Now we can begin to answer our questions. What's the difference between a healthy cell and a cancer cell? We'll answer this question by looking at gene expression, or which genes are on or off in cancer cells as compared to healthy cells. We will do this by measuring the types of mRNA found in both types of cells. First thing we need to do is find some cells. We will collect mRNA from melanoma, a kind of skin cancer cells, and healthy skin cells from the same patient. This way we can look at differences in gene expression in cells that have the exact same genetic blueprint. Of course, only physicians and qualified scientists working with the physicians would be permitted to collect and use such tissue samples, but for this virtual lab, we'll put on our virtual doctor caps. The patient has arrived. Let's get started. We will collect a healthy tissue sample and a cancer tissue sample from our patient. We'll need a scalpel and 2 empty sample tubes. We're going to assume they sanitized when they did that. We can extract the RNA from both tissue samples by dissolving them in a mixture of various organic solvents. We could separate the DNA, proteins, and other cell components, leaving only RNA in solution. Drag the pipette down and add some solvent to the healthy tissue sample. Next, we'll mix our tissue samples on the vortex. As the vortex mixes the samples, the tissue will dissolve and the RNA will be released. Place the tube containing the healthy tissue sample onto the vortex. The Samples have been placed into the centrifuge. The centrifuge will spin the samples and separate the RNA from the rest of the cell contents. So there's a little side question here. Why doesn't the DNA stay in solution? The strands in DNA are much, much longer than RNA molecules. Remember that the centrifuge uses centrifugal force to push the heavier things down to the outside, in this case, the bottom of the tubes. So the heavy stuff will stay at the bottom, while the light stuff will stay floating. We are going to still just that top phase. We'd want to switch tips because we need to show which are healthy and which are cancerous. We don't want to have our samples get mixed together. The samples contain several different types of RNA: messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. Our mRNA molecules, but not tRNA or rRNA, always end in the sequence of adenines, known as the poly A tail. Only the mRNA reflects gene expression, so we'll save the mRNA and get rid of the others. We'll wash our RNA samples over columns filled with small beads that will only bind to RNA strands that have a poly A tail. The other molecules will wash away. So we see the tRNAs and rRNAs flow through. The mRNAs stick to those beads that have strings of T's. The T's and A's will stick together. Our columns now contain strands of mRNA attached to the poly T beads by their poly A tails. Now we'll wash a buffer solution over the columns to detach the mRNA from the beads. We're going to see this done because base pairing between DNA molecules is affected by chemical conditions such as salt concentration and pH. The chemical conditions of this buffer do not allow for hybridization. Thus, the mRNA separates from the poly T beads. We're basically adding enough salt that it's going to disrupt the hydrogen bonds. Next, we'll make a DNA copy of the RNA. As we do that, we'll give the DNA some color. We'll add some labeling mix to our 2 RNA samples. We'll use the green labeling mix for the RNA from healthy cells. For the cancer cell RNA, we'll use a labeling mix containing red fluorescent dye. The labeling mix will assemble a complementary DNA strand opposite every RNA molecule. Why do we have to make a cDNA copy that's complementary DNA? Why not just use the mRNA? When we make a cDNA copy of our mRNA, we incorporate a fluorescent label in our cDNA molecule. This label will allow us to visualize the cDNA later on. Also, DNA is much more stable than RNA. The labeling mix contains poly T primers, which bind to the poly A tails on the mRNA, usually referred to by the scientific community as oligo-DT primers. Reverse transcriptase, an enzyme which will synthesize a strand of complementary DNA, or cDNA. And labeled nucleotides, which will be incorporated into the new cDNA molecule. The nucleotides in this tube have a green fluorescent molecule attached to them. This is just like your normal PCR would do during the elongation phase. Reverse transcriptase has assembled the labeled nucleotides into the cDNA molecule, and the mRNA molecule has been degraded. We're going to do the same thing with red for cancer. Good work. Our tubes no longer contain RNA, but now contain complementary DNA. The healthy cell cDNA molecules are green, and the cancer cell cDNA molecules are red. This is what we will apply to our microarray. Two complementary strands of DNA can separate and rejoin. Two complementary DNA strands from different sources can pair with one another. This process is called hybridization. In the next step of our experiment, we'll take advantage of a key property of DNA, complementary base pairing. If two complementary DNA strands are mixed together, they will soon find and base pair with each other, reforming a double-stranded DNA molecule. It doesn't matter where they come from. They'll do that even if they were not originally paired. Scientists call this hybridization and is the key to how DNA microarrays work. Stuck to our microarray are little piles of single-stranded DNA molecules. A single spot contains many identical copies of the same gene, and each spot on the microarray represents a different gene. So you can see here that the same DNA sequence is going to be within each spot, but if you compare two spots to each other, they would be different. Our computer database lists which gene is contained in which spot. Now let's add the single-stranded cDNA we prepared from our healthy and cancer cells to the microarray. Remember that all the cDNA molecules in our tube represent human genes, and for every molecule of cDNA, there's a matching spot of the single-stranded DNA on the microarray. We're putting on the healthy right now. And we're going to put cancer. Let's take a closer look at what's happening on the surface of the slide. Most of the cDNA molecules have hybridized to their complementary DNA strands on the microarray. A few stray cDNA molecules did not hybridize. So we have some unhybridized ones floating away. We have green ones in one spot, red ones in another spot. And if you have red and green together, you end up with yellow. We're going to wash off the extra cDNAs that didn't bind. And we're going to analyze the data. So we're going to use a microarray scanner. First, we'll scan the microarray to see where the green labeled cDNA from the healthy cells bound. To simplify things, we'll examine just a section of the microarray. Many of the spots are green, but not every spot. The dark spots are genes that were not transcribed in the healthy cells. Remember, not every gene gets expressed in every cell type. Now we're going to scan for red. Lots of spots again, just as for the green genes, but the red pattern looks different. It's clear that skin cancer cells and healthy skin cells have different gene expression patterns. But how can we sort through all this data to make sense of it? The microarray scanner can merge the red image with the green image and display a composite picture. Here are the red and green spots merged together. Any spot that contains both red and green cDNA shows up as yellow. A yellow spot contains a gene that hybridized to both green and red cDNA, which means that the gene was expressed in both cancer cells and healthy cells. Genes that show up as yellow spots probably aren't very interesting to us since their activity doesn't change when the cell becomes cancerous. But look at all the red spots and all the green spots. And for colorblind people, you can tell the software to make the spots yellow and blue instead. Red spots show genes that red spots show genes that produce more mRNA in the cancer cells than in healthy cells. They're turned up in cancer. Green spots show genes whose expression is turned down in cancer cells. You are a researcher interested in studying genes that have increased level of activity in skin cancer cells. What color spots should you choose to study? Well, based on what we just saw, we want to see where red is more active. That's right, red spots represent genes that are turned up in cancer cells. Wow, that's a lot of red spots. Have all of these genes gone bad? No, many of these red genes are normal. They're red on our microarray because they are controlled by a gene that has gone bad. Gene 4263 is one of the genes that are turned up in cancer cells. Gene 4263 produces a protein product whose role is to turn down the expression of several other genes. What color are the spots that are turned down by gene 4263? So we're going to go with green. Yes, gene 4263 turns down a set of genes which are represented by green spots on our microarray. These are genes that were on in a normal healthy cell but are now turned off in the cancer cell. So since all the red is gone, it's not going to be yellow, it's just going to show the green. Gene 6219 is normally turned on in skin cells. In our cancer sample, this gene is defective. While the gene still produces mRNA, the defect prevents the mRNA from being translated into protein. What color is gene 6219 on our microarray? Well, since it's going to still be producing, its expression didn't change. Because remember, we're not measuring protein levels, we're measuring mRNA levels. Good job. Gene 6219 is represented by a yellow spot on our microarray. While RNA expression usually indicates protein activity, this is not always the case. In this example, gene 6219 mRNA is made of both healthy and cancer cells, causing it to appear yellow on the microarray. However, in cancer cells, a defect prevents the gene from being translated into protein. Because gene 6219 shows up as yellow, the defect cannot be detected using microarray analysis. This is a major limitation of the technology. So how can researchers know if a gene is producing protein? Scientists rely on a technique called protein expression analysis to tell them if the mRNA is being translated into protein. DNA microarray analysis is a powerful tool that can identify genes that are expressed differently in two different cell types by comparing every single gene in a single experiment. That's pretty impressive. However, as we have seen, DNA microarray analysis does have limitations. DNA microarray analysis on its own cannot tell you which genes or genes went bad to cause a disease, cannot cure a disease, and cannot identify every gene that is behaving inappropriately. All right, since you can pull up this page, you can go through and read the rest of this information on your own. We don't need to do that in the video.